Okay, this is the Professor, CBMD PhD, and today's Thursday, August 17th, 2023. It is 12:44 and 1 second, a.m. Eastern Standard Time, and today's research, bioengineered tools. So let's jump to it here. As bioengineered tool, unmasked cancer cells, date August 10th, 2023. Source, Stanford University. As sugar-coated proteins, called mucins, are implicated in many diseases, including cancer, a team has now bioengineered an enzyme-based scissors that selectively cuts mucins of cancer cells, removing their cloak of protection from the body's immune system. As mucins are sugar-coated proteins whose primary function is to defend the body against physical insults and pathogens, but cancer cells can co-opt mucins to aid their survival. As cutting mucins up cancer cells is a plausible therapy, but mucins exist in various forms on every cell in mammalian bodies, so targeting mucins indiscriminately could have unforeseen side effects. The solution device by scientific investigators in the microbiological team is essentially an enzyme-based scissors composed of a mucin, a protein-cutting enzyme called a protease. That specifically cuts mucins fused to a cancer cell, targeting nanobody, an antibody fragment. This two-part biomolecule selectively targets and prunes only mucins associated with specific cancer cells. This study carried out in laboratory-grown human cancer cells and in mouse studies that simulated human breast and lung cancer, found the biomolecule treatment significantly reduced tumor growth and increased survival. Their findings, nature, and biotechnology have broad applications, as mucins are associated with many diseases, including cystic fibrosis, respiratory diseases, and viruses. They found that they can target the mucinase that cancer cells use it to remove mucins from those cancer cells, and there was a therapeutic benefit. Although cancer cells use mucins for nefarious purposes, mucins are generally good, but when mucins go bad, they are awful. Mucins play important roles throughout the body, such as forming mucus in our gut and lungs and protecting us from pathogens. Cancers dial this natural process up to 11, hijacking the functions of mucins to protect themselves and spread throughout the body. This study investigated two functions of mucins associated with promoting cancer progression. The first helps cells survive in free-floating, low-adhesion environments. As cancer metastasizes and spread in the body, cancer cells break off, float to another part of the body, and take root. Traveling cancer cells need to survive in low-adhesion environments. Most cells cannot, but cells that have been modified by mucins can. The second function is to bind the checkpoint receptors, which are essentially immune system guard dogs, and inspect cells in the body. Some cancer cells accessorize their cell surfaces with mucins that are coated with specific sugars that bind particularly well to these receptors. When this sugar-decorated mucin binds to checkpoint receptors, it indicates the cancer cell is not a threat and blocks the body's immune response. This causes immune cells to ignore the cancer rather than destroying it, as they should. The researcher's mucin-seeking biomolecule is made of two parts, fused together. The first part is bacteria-derived mucinase that cleaves mucins. The second part is a cancer-specific nanobody that binds to a corresponding antigen on cancer cells. The research team has extensively studied bacterial proteases that cleave mucins. These mucinases cut when they encounter specific arrangements of peptides, amino acids, and glycans, sugars, and mucins. For this study, the researchers chose a mucinase called STCE, pronounced sticky, derived from the bacteria E. coli. As bacterial enzymes are already used in treatments for cancer, such as childhood acute lymphoblastic leukemias. However, mucinases haven't been tested as injectable therapeutics, and so the team needed to verify the STCE mucinase works, and is safe. The team tested the STCE mucinase in mice and found it works when it ravaged mucins throughout the body, verifying the target to target the mucinase, the tumor-associated mucins. As previous research by the scientific investigators, a research laboratory, and others demonstrated that fusing antibodies to enzymes can target their activity to specific cells. It requires engineering the enzyme to work a little less well, so it only cuts but near the target. Many mutations of STCE mucinase later, the team created a version called STCE Engineered Sticky that met their needs. The team selected a nanobody known as 5F7 for their biomolecule because it is well studied and it corresponds to the antigen called ATR2 associated with breast, ovarian, and other cancers. The researchers assigned two different orientations of the STCE mucinase ATR2 nanobody combo and tested each for yield, stability, mucinase, activity, and binding ability. The best performing orientation was nicknamed Alpha ATR2 ESTCE. Next, the researchers tested the Alpha ATRT ESTCE biomolecule to see if it selectively killed the target cancer cells in a series of test laboratory dishes. Then they verified the biomolecule worked and was non-toxic in two different studies using mice. The first of these experiments simulated metastatic spreading lung cancer, and the second simulated human breast cancer as tumors located in the breast region of mice. And these scientific studies showed that alpha ATR2 ESTCE biomolecule was effective on both mucins on tumors and metastasizing cells. In the studies in mice, the researchers found that the alpha ATR2 ESTCE treatment significantly reduced cancer growth and increased survival compared to the untreated group of mice. And so how close does this study bring us to a new cancer therapy for humans? Closer, but not there yet. One major step is to see if they can make a comparable targeted mucinase using a protease derived from humans. The one in this study isn't derived from humans, and so it has a higher risk of unwanted immune response. Although more research is needed, this study represents a big step forward in cancer research. They have decades of evidence from cancer patients and experiments that mucins are important in cancer. There was not that much that they could previously do to get rid of these mucins. They were inspired that they finally had an approach to degrade mucins on cancer cells. And this is the professor of microbiology, bioinformatics, pathophysiology, and teacher of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.